Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky textbooks, revision guides and practice papers. Everything you need to learn, review and prepare for your senior phase assessments. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. You can now also collect Young Scot reward points by using the codes within the description of each episode to claim discounts and rewards on their website, www.young.scot.
Actually, some individuals are born with an external tail, so their coccyx is longer.
The haploid human genome consists of approximately 3 billion nucleotides, but changes in a single base pair can cause devastating physiological malfunctions. We refer to these changes in DNA as mutations. Without mutations, however, evolution would not be possible, as beneficial adaptations would not have developed upon which natural selection can act. Without mutations, you simply would not exist. In this podcast, we will consider the effects of both single gene and chromosome structure mutations and the impact these can have in individuals, as well as the importance of mutations in terms of evolution. To help us understand more about this key area, I am joined by scientist and author Nathan Lents from New York. Hi, Nathan.
Hi, how are you?
I'm good, thanks. How are you?
We're hanging in there. I am a professor of biology at John Jay College, which is part of the City University of New York. So it's a public college in the city of New York and has 26 campuses spread around the city. It's the fourth largest university system in the United States, even though it's only in one city, but it, of course, is our biggest city. So my work is I'm involved in several different kinds of research projects, but the majority of my research is focused on human genome evolution. So how the human genome has sort of come to its current state, how it operates, how it functions, where the genes are, what the genes are. And mostly I'm interested in the differences between humans and our closest living relatives, which are the apes, the African apes, and the other African apes, I should say, because humans are indeed African apes. We meet that criteria of our species evolved almost entirely in Africa over the last 15 million years. And we are apes, right? We are primates that don't have a tail. So I would say that my work is interesting because suddenly in the last few years, we have access to a different set of human relative. When I thought of humans' closest relatives, when it comes to studying DNA, the apes would be all that there is. But actually, we now have sequenced genomes from the Neanderthals and another curious species called the Denisovans, or sometimes pronounced Denisovans, depending on what part of the world you're from. And these are a strange population from whom we have no skeletons, but we do have an entire genome, if that's not interesting enough for you. So we have, it's funny to me that my line of work now involves studying organisms that it wasn't even possible to study just a few years ago. And that's science for you, right? It's always pushing forward and discovering new things. So I am interested in human ancestry and how we got this way. And in fact, I would say the question that drives me most is, how did humans get this way? Compared to how different we are to the other apes, we're very similar underneath our skin, but at the same time, we write poetry and build skyscrapers, and we don't have any idea why, like how that evolved, because we can think about the ways that we're different from the apes in terms of our anatomy, in terms of our behavior, but the genetics of it, which we assume drives everything else, we don't actually know. We don't know how humans got so different from chimpanzees, for example, which are closest relatives? So those are the questions that drive me.
The young people in Scotland, they will recognise the genome of an organism as its entire hereditary information that's encoded in DNA. And the genome, of course, is made up of genes and other DNA sequence that don't code for proteins.
Right. So DNA is thought of as a very static molecule, and in fact, in your lifetime, the DNA in your cells is pretty static. It's pieces of information, and that's it. It's very much like a book, and a book has no life of its own. It can't do anything. But when you read that book, you learn things, and you gain that information from the book, and then you can go do things. So DNA is very much like that. It's like the book. It's like the instruction kit for building an organism, in this case, a human. But it can't do anything on its own. Everything that DNA does or is claimed to do has to be done by other molecules because it's just simply the information in a coded format. But it's actually even less than that in a sense because very little of the genome actually does code for anything. About 3% of the genome codes for proteins, and that's the number people often toss around is only 3% of our genome is functional. That's not true. Only 3% codes for proteins, but another about 25% of the genome is the instructions on how to use those 3%. And in fact, we have more instructions about our instructions than the instructions themselves. So if the instructions are how to build a protein, that's the easy part. How to build a protein in terms of coding the information. Very easy to code how to build a protein. But what's much more complicated in the case of humans and other higher animals, higher organisms, is that when to build that protein, where to build that protein. Those are much more complicated pieces of information that have to be coded in the genome. So the genome is a little bit of it codes for proteins. The rest of it codes for how to use those proteins, when, where, how to build them. And then the other 75%, which I haven't talked about yet, is more or less junk. It's pieces of information that aren't doing anything, at least aren't doing anything useful. Sometimes it's active in negative ways. However, that DNA that's unuseful now might very well become useful in the future. Much of it was formerly useful. Some of it was formerly part of a virus, actually. So viruses have DNA or genetic material, kbRNA, and that gets inserted into our genomes and sometimes in a permanent way that we then pass on. So we carry a lot of, I refer to them as carcasses of viruses in our genomes, in our DNA, in our chromosomes. So we have about 9%. Sometimes people say 8%, but 8 or 9% of our genome comes from viruses. So it's a hodgepodge. It's a mismatch of lots of things. Now, I want to return to a point that I made about DNA being static. It's static in your body, in my body, in our cells, for the most part. There's a couple of exceptions to this. Immune cells, certain cells in the immune system, certain T cells and B cells can rearrange their DNA in order to make different kinds of antibodies, which is really an interesting thing to think about right now during the COVID-19 pandemic. But in general, those are the exceptions. Every other cell of your body has the same DNA that it was sort of born with, that you were born with, in terms of the sequences, barring a couple of mutations, which I assume we'll talk about that, but those are almost never going to be of consequence to you. So it is static in your lifetime. But I studied the way that DNA changes over hundreds of thousands of lifetimes, of generations. And if you think about DNA in those timescales, it is anything but static. DNA is very mobile and it's very subject to change. It's very dynamic, as we say. Otherwise, the first organisms that had DNA, we'd all still just be that, and there wouldn't be any change. So actually, over time, DNA is known for its ability to change. Some of those changes are mistakes, and others, there's all kinds of different ways it can change. But if you look at your DNA and compare it to that of chimpanzees, the first thing that will strike you is the similarity. But the next thing will strike you is that the differences are complicated, and they tell us that there's a lot going on over evolutionary timescales. That the DNA isn't just sitting there waiting to be read, but actually pages can be ripped out, pages can be duplicated accidentally, pages can be rearranged. Even the text on a page can be rearranged. So it's not just a perfectly faithful copying process. That it's actually quite error prone and dynamic and can interact with other organisms such as viruses. So hopefully that wasn't a tangent for you, but it's setting the stage for our conversation.
It does indeed, and of course, talking about books and pages and all the rest of it, you are of course an author, and you've got your own book out called Human Errors: A Panorama of Our Glitches from Pointless Bones to Broken Genes.
So Human Errors is a book all about sort of flaws, glitches, quirks, whatever you want to call them, in the human body, from our DNA, from our cells, all the way to our organs, even to our psychology, the way we think and act. We actually have quite a few. And these aren't just limitations. Everything has limits, but this is beyond that. These are like some real sort of glaring defects in our physiology and in our genetics that really call out for an explanation. And the reason why I wrote a whole book about it is that it's not just like encyclopedia entry of these things. It's a story, it's a narrative, because each one of these flaws has an interesting backstory, has an interesting history, and it actually reveals something about our past, about how we used to be, how our ancestors were, and things that happened to them are captured in some of these glitches and flaws. So when we think of studying human evolution and studying human ancestry, we tend to think of fossils and bones and things like this. But actually, you can study your body as it is right now and find clues of our past lives. So that's what the book is about. And it's not just the summary of this, by the way, the conclusion, if I can give it away, probably shouldn't too much or you won't read the book, but is not that we are just this ugly, dirty, non-functional sort of crappy bodies. That's not really what I mean, even though I do argue, and I think forcefully, that we are the most flawed of any animals, or at least any vertebrates. But that's not the lesson. The lesson is why are we allowed to be so flawed and so glitchy? It's because we've become very good at overcoming our flaws, and we overcome our flaws with our intelligence, but most importantly with each other. Right? We have an intricate social structure, so you don't have to live and die based on how good your body works, because we invent things, first of all, to overcome those, but we also work together and we can compensate for one another. If your arms aren't any good, you just do a different line of work that doesn't depend on that. So I'm oversimplifying. So the sum of the story, or the conclusion of the story, is that aren't you glad we have these glitchy bodies? Because it's a sign that we're not just living by the standard laws of natural selection. We're actually working by cooperative evolution, sometimes called cultural evolution. So that's the summary of the book. Now, how mutations in DNA factor in is there's one specific chapter that's all to do with the genome itself and the funny, weird, glitchy things that we have in our genome. And I hope we can talk about some of those. But also throughout the book, there's a lot of genetics in there, because ultimately it has to come back to genes, right? So if our heart isn't working very well under certain circumstances, it's because the genes have been allowed to sort of fall into disrepair. We're ultimately a genetic species like everything else. So genes factor all throughout the book, but I have one chapter specifically on what I call junk in the genome.
It's worth noting that in genetics, the term junk DNA refers to regions of DNA that are non-coding. A genome is made up of genes and other DNA sequences that do not code for proteins. DNA sequences that code for protein are defined as genes, while other sequences regulate transcription and others are transcribed but never translated. And you can learn more about those processes within the gene expression key area of the higher course. For now though, let's start to take a closer look at mutations, which are changes in the DNA that can result in no protein or an altered protein being synthesized.
Right. So there's all kinds of different mutations. There's different ways you can get the mutations, but also different effects of those mutations. So as you mentioned, sometimes a mutation can actually alter the shape or structure of a protein by switching out an amino acid. So proteins are built of amino acids. Those are the units. It's like beads on a string. And the individual beads are amino acids, and there's 20 different beads, 20 different amino acids, but then they get all folded up into a complicated shape. They're not just beads on a string. However, that's coded for with the DNA. Well, if you make a mutation in one specific base of DNA, you can change what amino acid is at that position. And if it's at a critical position in the protein, it'll change the shape of that protein. And if you change the shape, you change the function potentially. You can make it work slightly better, slightly worse. You can make it, if it's an enzyme, it might go for a slightly different target. Or some target that it only weakly works with, it'll work better, or a binding partner. There's literally hundreds of possible effects when you change out a single amino acid. But the other thing that you said was change a protein. You could also lose a protein through mutation. And you could lose a whole chunk of DNA, so a whole gene goes away, and therefore so does its product. But you could also, a single base pair change could destroy a protein by preventing it from being made in the first place. So you can insert a premature stop codon, and I don't know if you'll cover that with your students, how that works. That's one way to do it. Or you could prevent something in the regulatory sequence that prevents it from being turned on, so prevent a transcription factor from turning a gene on. Usually you need more than one base pair change for that, but just by example. So there's lots of ways that you can destroy or alter protein function through the tiniest little changes of DNA. But I, in my lab, tend to study bigger changes in DNA that happen over time. So whole chunks of the genome can be copied accidentally or duplicated, we could say. And that can lead to multiple copies of a protein, or it can lead to, sort of weird fusions. Part of this gene ends up now linked up with another one, and that's a brand new gene that gets made, and things like this. So over time, what seems like very unlikely events become not just likely, but probable, because every time a human cell divides 1 into 2 is an opportunity for one of these mistakes to be made. And then become permanent. And if it's unlucky enough or lucky enough to happen in a germ cell, which is a cell that's going to give rise to either a sperm or an egg cell, then it might be passed on into the population. People often forget that. That's a very, very, very important point about mutations. All the mutations that you're ever going to experience in your life are just yours, and they will die with you. But if it happens in a gonad, in a germ cell, that's the only way that a mutation will actually get passed on to the next generation, is if it's in a sperm or an egg, if it ends up in a sperm or an egg. And often these mutations have nothing to do with how sperm or egg work. So one thing I always remind students is, like, when a mutation first happens, it actually doesn't affect the person who experiences the mutation. It affects their children, right? They experience a mutation in a cell that then gives rise to the next generation. And that's who gets the mutation and experiences the effects of it, not the person in which it actually happened, unless it happens to affect the sperm or the egg, which would be a very small number of them, but it does happen.
As Nathan's explained, mutations are random, spontaneous, and rare errors in genetic information. Within the higher course, we need to be able to name single gene mutations and describe their effects.
So you can affect a single gene by just changing a base pair of DNA or making a small insertion or deletion or something like that. But when you have what we call gross chromosomal changes, these are most commonly the result of errors in meiosis. So meiosis is the process in our germ cells of sort of shuffling around the DNA material, the genetic material from our 2 copies of chromosomes. So I have to back up a little bit for this to make sense, but we have 2 copies. I shouldn't say copies. I should say we have 2 versions of every chromosome. So they're not copies of each other. They have the same kinds of information, but different versions of that information. Think of them as like having different versions of a textbook. From one year to the next, publishers will come up with new editions all the time, and we can talk about why they do that in some other podcast. But almost all the same information's there, the same chapters. It's mostly the same, right? But there's slightly different versions of it. Well, you have those 2. You have 2 versions of every one of your chromosomes. One you inherited from one parent and one from the other, biological mother and father. Well, right before you then make your gametes, sperm or egg, in order to possibly pass them on, you shuffle that information among what you got from one parent and the information you got from the other parent. The reason why that shuffling is important is that you don't want genes to be linked together. You don't want them to be inherited as pairs or more than pairs, but whole groups of genes. You want all of your genes to be inherited separately so that they can be acted on by natural selection as individual units rather than clusters of units. Anyway, the point is that during meiosis, which is the process of making sperm and egg, the information from your maternal and paternal DNA gets all shuffled up. Well, that is a very, very error-prone process because it involves lining up DNA, sliding strands from one to the other, and then crisscrossing it, and then unwinding it again. And that causes errors, duplications, and even sometimes breakage. You can actually break the chromosome. That's sometimes called catastrophe because it usually condemns the cell to death. However, sometimes those errors are fixed, and they're fixed with sort of improperly. They don't go exactly all the way right back to how it once was. And this can be passed on. So the most common way this is just by simply strand slippage during that crossing over, and you get duplicated whole chunks of chromosomes. And that sounds like it would be a good thing. And in often cases, it's actually neutral, but it can actually be a bad thing because you are programmed to have the sort of a set dose of every gene. You need this copy of these genes, and that's it. You can't just add extra genes. However, occasionally, if it's subtle enough, you can add extra copies, and it's a good thing, potentially, because it gives you backup copies that you can then play around with in terms of evolution by natural selection. But so gross chromosomal changes involve duplications or deletions, actually, of whole chunks of chromosomes. And then small-scale changes are usually due to the DNA copying apparatus encountering some kind of mistake there.
What can you tell us about substitution mutations and the difference between missense, nonsense, and splice site? How would you explain that?
So substitution refers to what nucleotide is used in the DNA. So you're substituting an A for a T or an A for a G. It's basically a copying error. So when the DNA strands separate from each other and then are copied, sometimes mistakes can be made. And when these mistakes are made, we call that a substitution. A lot of times they're fixed, by the way. So most of the mutations that are originally made are actually fixed. So I shouldn't call them mutations. By definition, mutation is it didn't get fixed. So it's a mistake that becomes a mutation when it is not fixed. Well, a substitution, depending on where it happens, can have different effects. Just like a spelling error in the English language can have different effects, right? So if you have the word talk and you change the first letter from a T to a W, you make a different word, and that word is walk. And then if you delete the final letter, right, talk becomes tall or whatever. So it's the same thing with DNA. Where the substitution happens can affect what the effect is. So if you change one nucleotide of DNA from one to another, you can change the amino acid. You change the codon. That's a missense mutation. So a missense mutation is a changed amino acid from one to another because of a substitution in the DNA. Another effect you can have is called a nonsense. And nonsense means it's like a subset of missense, where you do change the amino acid from one thing to another, or the codon from one to another. But instead of going from a regular amino acid to another regular amino acid, you go to a stop codon. So it's actually not an amino acid at all. It's a nonsense codon, which means nonsense means no code. Sense means code. When you're talking genetics, sense, the word sense, you can substitute the word code. So missense means wrong code. Nonsense means no code. So the DNA triplet doesn't have an amino acid. It goes for stop. Now, a splice site substitution is so splices are what is the process of merging exons, stringing them together to make a final protein. So remember, we have exons and introns, and that's DNA, continuous DNA that's exon, intron, exon, intron, exon, intron, and then you splice out the introns, meaning cut them out of the messenger RNA and then fuse them together, the exons together, to make a continuous thing. Well, if you get a mutation right at the spot where exons and introns are fused together, you can actually prevent or create a splice event. And then you don't make the same protein anymore because you either are missing an exon or you're adding a little bit of extra sequence to an exon, and that's a splice site mutation. But what happens to cause it is the same, meaning a substitution of an A for a G or a C for a T. Whatever it is, you're changing the DNA sequence by one base pair change, one base pair substitution.
And obviously, you mentioned that single gene mutations are fairly small mutations, but they can obviously have some quite big impacts on humans who may be unfortunate enough to have those mutations. And I'm thinking about examples such as sickle cell disease, which would be an example of a missense, PKU, another one, and then even things like Duchenne muscular dystrophy being an example of a nonsense mutation. I mean, these are quite significant.
Right. Most of what we know actually about human mutations and genetics comes from these diseases, the ones you've mentioned. That's, in fact, actually, if you read the study of genetics, the history of the field of genetics, you're really reading a history of research on genetic diseases. I mean, that's how we sort of figured out what genetics is by studying diseases that ran in families. because that was confusing. As why would this run in this family? Um, and yeah, sometimes the reason is a single base pair change. So sickle cell anemia, there's a single base pair change, which leads to a single amino acid change that causes a misshapen, or I should say, altered folding of the hemoglobin molecule, and then it doesn't pack into red blood cells as well, and therefore the red blood cells have a slightly different shape, which under normal circumstances can be okay, but during exercise or strain, it doesn't fit through the capillaries as well and starts to get stuck and everything else that comes from that. So yeah, single base pair change. Cystic fibrosis, same thing. A single base pair change causes this, up until recently, fairly lethal genetic disease. I say up until recently because people are starting to survive longer because of treatments and also because of double lung transplants. However, I predict in the next 10 years or so, we'll begin curing people with sickle cell and cystic fibrosis and so on, because it is so easy to fix a single gene when you know everything that we know about it. Bigger genetic issues are going to be much harder to fix. But single locus is sometimes what that's called. Monogenic traits or monogenic diseases are due to the most subtle possible change in the DNA code.
And cystic fibrosis, that's caused by a frame shift deletion.
So there's multiple alleles of that. So the gene in which this happens, there are, when I say allele, that means, the version of the gene that leads to the disease. And these run in families, of course. And what we've found is that there's actually multiple ones out there, and it can. And in fact, two families. We used to always think if there's cystic fibrosis in two families, if they intermarry, you have greater odds. But actually, if their versions of the alleles are different, then there actually isn't an elevated risk, because you have to have both of the same bad copy in most cases. It depends. Again, it depends on what the allele is, right, and where the misfunction is. But yeah, so frame shift is one, and frame shift usually leads to a drastic change, because what happens there is everything in the code is then off. So a frame shift, because the code is written in triplets of 3, right? So if every 3 base pairs codes for an amino acid if you're in frame, starting from the start codon. And so if you shift that by just 1 base pair or 2, then everything will be off. Every amino acid will be wrong. Even though you've only changed the mutation or changed the sequence in 1 base, everything is then incorrect after that. So those tend to be fairly drastic. They tend to be what we call null alleles, where it would just destroy the function of the gene entirely.
And those frame shift mutations, because of the way they work in shifting everything, those are obviously caused by either insertions or deletions.
Insertions or deletions of 1 or 2 base pairs. If it's 3, then it's not, right? It doesn't change or a multiple of 3, because you can have insertions of 5 base pairs, 8 base pairs. But if it's 3, 6, 9, or 12, then you're fine in terms of the reading frame. You might still alter the protein, but you're fine in terms of the reading frame.
And within our higher biology course, within the course specification that's provided by the SQA, one of the study activities that's suggested is to go away and look at some of these human conditions that are caused by these mutations. And I think one of the things, as you mentioned, people maybe need to realise is that actually by going and studying those, you can open that up and really look at the history of genetics, never mind those specific conditions.
Right, exactly. And in fact, Britain is where a lot of the earliest work on genetics was done. And some of the scientists were following these diseases where infants were born with major problems, usually major problems, but it would pop up in families repeatedly. And so some of the physicians working mostly in London, but in other places in Britain as well, they were able to sort of track this down. And so a history of genetics research is really a history of genetic diseases and our understanding of them. And that was when the big breakthrough happened, first in Britain and then in America and then in Germany. Those are the powerhouses of early genetics research. But this was often before we really even knew how genes coded for proteins, right? So that stuff was still being worked out. So genetics almost had an air of magic to it, that we knew that there was something that was inherited and traits ran in families, children looked like their parents, things like that. But how was still being worked out.
And obviously, we've spoken a little bit there about single gene mutations. One of the other main ones that we look at is chromosome mutations or chromosome structure mutations. How would you describe the difference between single gene and chromosome structure mutations? How would you explain that to young people?
Well, it's mostly a matter of scale. So the single gene mutations are almost always due to what we said, copying errors, where you have a small insertion or deletion or a single base pair change. But you can also have changes to the gross in this case means size. So a large scale alteration of a chromosome, where part of a chromosome breaks and then is repaired with some duplication or I should say deletion or insertion happening, where whole chunks of the chromosome can be lost or whole chunks of the chromosome can be duplicated. That usually occurs in meiosis. You can also have translocations, where parts of one chromosome, break off, and then instead of being put back in the right place, they're attached to a different chromosome. And you would think, well, that's fine. You still have all the same material. But the problem is the local area in which a gene exists in the DNA actually affects how that gene is expressed. So if you take a gene from one place in the DNA and put it somewhere else, even if it's still intact, it'll be changed in terms of how it works. When, where, and how much it's expressed will be altered. And in fact, the famous example of this is the Philadelphia chromosome, so-called because it was discovered in the city of Philadelphia. Well, this is a hallmark of a certain type of cancer, a certain type of leukemia, where part of one chromosome is flipped with part of another. So all the same genetic material, it's just been rearranged a little bit. But what that has done is causes complete dysregulation of one of those genes. And it turns that gene into an oncogene, which is a gene that causes cancer, in this case, leukemia. So all the same genetic material, you've just rearranged where that material is found in the genome, and it causes a drastic change in the expression of a key gene, which leads to cancer. So our genomes are fragile in that way, that just tweaking them usually has negative consequences. Now, the genius of evolution, of course, is that you're tweaking them over long periods of time, meaning, yes, all of this negative things are happening, and then the sudden, rare, good change that happens by pure luck is allowed to flourish and take over in the population. So those are rare events, but if you're talking about 100,000 generations, something that's rare becomes probable.
When we talk about chromosome structure mutations, we would tend to talk about duplication, where a section of chromosomes added from its homologous partner, deletion, where a section of chromosomes removed, inversion, where it's reversed, and then translocation, where a section of the chromosomes added to a chromosome but not its homologous partner.
Right.
Where would the Philadelphia chromosome mutation?
That's a translocation. That's actually a reciprocal translocation. So what that means is you have a break, in two places, and the information is switched from one, the proper location of this gene, gene A and gene B, they get flipped. So it's like if I take you out of your home and put you in your neighbor's home, but I take your neighbor and put him into your home. So all the same people are there, and the homes have the same number of people, but the identity has been switched, and that's what the Philadelphia chromosome is from.
That's going to cause a little bit of chaos in the house if that happens.
That's right, and it's a good example how you're still you, but you're in the wrong place, and so you can't really be you, even though you're intact, because you're not in your normal setting. It's a silly analogy, but it kind of works.
Yeah, I'm not sure my neighbour's wife would be too happy with that. In fact, I'm not sure my wife would be too happy with that either.
But you never know.
And it is a good example, because these chromosome structure mutations, they make substantial changes in the chromosome, which of course can be lethal sometimes.
Right. Yeah, it can. And the most common way this is lethal to the cell. And remember that we're a multicellular organism. So if this happens in the cells of your body, generally there's no consequence, because you just lose that cell and it's replaced by another one if it's a tissue that regenerates. And even if it's a tissue that doesn't regenerate, you have lots of extra cells in your body. You're going to be fine. So when we talk about lethality, you're talking about this happening in an embryo or in a germ cell. Anyway, the most common way that you have a lethal mutation is when you lose information forever. So if you break off part of a chromosome and it just does not get fixed, it does not get put back where it belongs, then that information is lost. In most cases, that'll have a negative consequence. You need your genes. Each one of them has an important function in your body. However, it might not be felt right away, right? So it might be a gene that really doesn't become crucial till later in life, for example. Or it might be a gene that's not necessary in all conditions, in all states. Like maybe that gene protects you against heart disease. Well, you might not be prone to heart disease because of other things in your body. Maybe you have a great diet and good exercise and lead a low stress life with lots of sleep. I don't know anybody like that, but in different times of our life, it could be that some genes that otherwise could be pretty lethal in different environments aren't. So the genes interact with the environment and the way that you live your life. And so different mutations can be felt very differently. Actually, the sickle cell anemia is a good example of this, where you can tolerate sickling of your cells in a lot of different environments, but at high altitude, you cannot. You cannot tolerate.
What do we mean by sickling, Nathan?
So sickling is what happens when the red blood cells are misshapen in this condition called sickle cell anemia, or sickle cell trait, sometimes called. So the hemoglobin doesn't pack in as tightly into the red blood cell. That's the root cause. Well, but a lot of times, the fact that the effect on the red blood cells, it doesn't fold over. So red blood cells actually fold in half when they go through capillaries. Capillaries are the tiniest blood vessels of your body, and if you look at it under a microscope, it looks like the red blood cell doesn't even fit. That's how small those vessels are, because red blood cells are tiny already. However, but they sort of fold over. If you can imagine taking like a donut and folding it in half, kind of, that's sort of what they do in order to squeeze the vessels. Well, if the hemoglobin is not packed in as tightly, they can't fold as well, and so instead they can get clogged, and they get these kind of funky shape actually under the microscope. Well, in a lot of cases, you're okay, because you have tons of redundancy in your circulatory system. You have lots more vessels than you actually need at any one time. And that's why you can tolerate having a wound and not, the whole area doesn't die. You've got lots of extra vessels around, and the blood will still circulate and get the oxygen where you need it. You have a lot of redundancy throughout your body. However, if you're at high altitude, you need every bit of that redundancy in order to deliver oxygen to the tissue. So if you're at high altitude with sickle cell anemia, you're going to experience altitude sickness very severely, and any kind of exercise will send you into a crisis and be lethal. So the same state can be non-lethal at sea level, but lethal at high altitude, for example. And it depends on how bad your sickle cell disease is in that individual. Same thing with strenuous exercise can bring out sickle cell anemia, so can high altitude. So and the kind of places where sickle cell anemia persisted for a long time were lower altitude environments, because as we in another story, sickle cell anemia also protects you a little bit from malaria. So malaria is a parasite that infects red blood cells, and they don't like the sickled ones. And so actually sickle cell anemia patients have a resistance to malaria. So it's persisted in some areas of the world because of its defense that it gives you against malaria. But none of those are high altitude places, because then if the malaria doesn't get you, the high altitude will. So it's a push-pull. And actually, evolution is a constant story of pushing and pulling, and forces of natural selection pushing us in different directions at different times under different conditions. It's never just a smooth, easy, simple condition.
And evolution is something that we'll come on to in the next episode. Obviously, the importance of those mutations and gene duplication in evolution is quite an interesting topic.
Yeah. So if you think about it this way, every gene in your body is performing some important functions, right? And if you hit that gene with a mutation, a random mutation, it's almost always going to be negative on the protein. And therefore, it's almost always going to be negative for you as an organism to lose some of your functionality through a mutation on this gene. But now imagine you have two copies. So it's become duplicated. So now you have two copies. And you hit one of them with a random mutation, and it's negative. So what? You still got the other one. You still have the other one. So what happens is these mutations don't kill the organism. They don't kill the gene. And so you're allowed to sort of tinker around. It's one example I used to use is let's pretend you had like an iPhone, and you use it to make your phone calls and your emails, and it's crucial. Well, if you open it up and start tinkering around with it, almost no matter what you do, you're going to hurt the thing, and you're going to lose your ability to do email and blah, blah, blah. Okay, but imagine you had two iPhones. Now you can open one up, and you can tinker around with it, And you don't lose your ability to do phone calls or emails or any of the other things because you have the other phone. And while you're tinkering around, yes, most of the changes you make will be negative. You might stumble upon an enhancement. And now you have the ability to switch over to the one that you've enhanced. So if 300 people out there have 2 iPhones each, yes, 299 of them are going to hurt their iPhone through their mutation, but one of them is going to be able to come up with something beneficial, and then that passes on. So that freedom to tinker really depends on you having 2 versions of it. And the good example of this is color vision in primates. So primates have slightly better color vision than all of the other mammals. Most of the other mammals have what is called dichromatic color vision. So there's only 2 kinds of color receptors or photoreceptors, color detecting photoreceptors, we'll say. So you can cover the other, so we're talking about dogs and cats, we'll say. They have 2 kinds of color detectors. They don't see all black and white. Some people think cats and dogs see in black and white. They don't. They see in color, but not as much color as us, as primates. So their 2 color detectors cover the best they can the electromagnetic spectrum of colors, the color palette, we'll say. Well, in the early history of primates, one of our genes for those photoreceptors got duplicated into 2, and it's in tandem. So it's on the X chromosome. It happens to be on the X chromosome. And it's a tandem duplication. So one copy got duplicated into 2 right in a row. And now you have 3 copies, right? Because you still have the other one, but then the 2 on the X chromosome. Well, those are now free to mutate and shift. And it doesn't just move the color spectrum along. You've got 3. And so our color vision is we cover much greater resolution in the color spectrum with our 3 color detectors than other mammals do with just their 2. So we are trichromatic. And this is basically all primates. I think it's super primates, actually. So it's primates and a couple of closely related organisms. And we have 3 color detectors. And that's why primates are so much more vision driven than smell driven. Unlike the vast majority of mammals, by the way, are driven by their sense of smell. That's why they have big snouts and not that great a vision. But primates, with our better color vision, we really evolved our way into a visual-based life. We navigate the world with our sense of vision, much less than our sense of smell. And so primates reduced our snout and brought our eyes forward for 3D, good, high-resolution vision. And that all was started by a duplication of a gene. A gene got duplicated, gave us better color vision, and so we evolved our way into making use of that.
And that's obviously one of your big areas of interest is the difference between humans and primates. One of the things that always jumps out for me is we've kind of got a tailbone, but we don't really, and you can still break it, and it's sore to sit down if you do, but we don't have a tail. It's kind of weird, isn't it?
That's right. You have the stump of a tail. You have part of a tail, and it doesn't really do anything for you, the coccyx. There's this idea of, oh, well, if you have a body part, it must be doing something. The coccyx really doesn't do anything for you. It does bear some weight when you're seated, but you got to remember, sitting is not a posture that we were designed to do, actually. Humans, the chair was invented much later in life, so early humans didn't sit. They squatted, they leaned, they laid, they stood. There wasn't a lot of sitting in most of our history. So the tailbone was totally superfluous. And we know that now because you can get cancer of the tailbone and you can break it. And if people remove their tailbones for either of those reasons, that's called a coccygectomy because the tailbone is called the coccyx. So if you have patients that have a coccygectomy, they have no trouble sitting. They don't have any trouble with like bowel control or anything like that. They suffer no side effects. You can remove the tailbone with no side effects. So that's one way we know that it doesn't really do anything for you, at least nothing crucial. But you don't just have a tailbone. You also have some muscles associated with it that if you did have a tail, you could flex that tail using those muscles. So the muscles are there, but the tail isn't. And it's just because, the tail regressed and evolution didn't really finish the job, so to speak, because it doesn't hurt you to have those muscles. They're not really doing anything bad. And so the pressure to remove those muscles was sort of lost. Would they probably continue to regress over time? Yes, that's genetic drift is involved in that. That's a longer story. But a lot of things we just sort of carry around in our body because they don't harm us, but they don't really do anything for us either.
Have you ever broken your coccyx, Nathan?
I haven't broken it, but I bruised it once pretty badly as a child. I was
I have flashbacks to doing something similar, and it's sore.
very painful. Yeah, when I bruised my tailbone, I was screwing around in my house. I was jumping hurdles and things, and I came down right on my tailbone, and I didn't walk right for a few days. It hurt very badly. Whereas if I had a softer landing, it might have been preferable. So I wish I didn't have a tailbone in that moment. But yeah, and actually some individuals are born with an external tail. So their coccyx is longer and they have they have more, some of the bones there. And what that sort of harkens back to, it's obviously removed surgically quickly, but what that harkens back to is the genetics of how we lost our tail. Because we can sort of undo that, and you can sort of see what that stump of a tail once looked like. But right now, a tail is a hindrance, so it's not if you're not using your tail for balance or grasping or anything, then evolution will get rid of it because it can get stuck and you get hurt. So it is a hindrance to have that. So there's this idea of if you don't use it, you lose it, and that is true in evolutionary time periods. Just because generally, if you're not using something, it can harm you by just being in the way.
Before we finished, I went on to ask Nathan about some of the other mutations that he found most interesting.
Oh, well, here's an interesting one that has to do with mutation as well. So sometimes a gene gets so mutated, it can start with a single event, but it can get mutated over time where it's not functional at all. And those are called a pseudo gene. So pseudo means false. So it's a false gene. Sometimes these are called dead genes. So these are formerly functional genes. And we still have them in our genomes, but they don't function at all. So think of it as like a broken down car in a junkyard. You can tell it's a car. It looks like a car. It has all the right parts, so to speak, but it cannot function as a car even a little bit. Like you'll never turn it on. You'll never be able to go anywhere with it. It's completely dead as a car. But it's not missing in action. It's there. It has all the parts. Anyway, so we have a couple of these that have some interesting consequences. One of them is our ability to make vitamin C. Has been lost because of a specific pseudogene, a gene that got mutated. And so you might say, well, what do you mean vitamin C? So vitamin C is a vitamin for us because we must get it in our diet. It's called an essential vitamin. We must get it in our diet. But actually, the vast majority of mammals don't need it in their diet at all because they make it themselves. If you think about your dog food and your cat food, there's no citrus fruit in there. There's no vitamin C in their diet, but yet they don't die of scurvy. So what's going on there? Well, they make vitamin C in their liver the way that most mammals do, most animals do. And we lost that ability because there was a gene, and it's called L-gulonolactone oxidase. For short, we call it GULO. The GULO gene got mutated in one of our ancestors, and we lost the ability to make vitamin C. And you might be asking, well, geez, if she or he lost the ability to make vitamin C, why didn't she just die of scurvy and take that terrible mutation with her and not subject the rest of the population to it? Well, the answer is if you were already just happen to have vitamin C in your diet, then losing the ability to make vitamin C yourself had no consequence. There's food that has vitamin C in it. We eat it now for its vitamin C, but it might have just been a part of the regular diet. And what I'm talking about are fruits. So fruits are rich in vitamin C. They're not the only places you get vitamin C, but they're rich in vitamin C. And so our ancestor, and it's not just us, this is true of, again, almost all primates have this need for vitamin C. Well, where do primates live? They live in tropical rainforests for the most part, and that's where you find a lot of fruit. So the diet is what allowed this mutation to not be lethal and therefore be retained by pure bad luck. So that's a good example of a mutation that should have killed us, and instead it didn't, and we inherited it all through bad luck, and now we're stuck with vitamin C as a requirement. And when I say stuck, I mean this is a major problem for our group of animals, because primates have always been restricted of where they can live in the world based on the availability of vitamin C. And every time they tried to go somewhere where a vitamin C poor environment, they died of scurvy, of course, without ever knowing why. And that's one of the reasons that Europe, the entire continent of Europe, was never colonized successfully by a primate until Neanderthals and humans. And we got scurvy when we did, because Europe is not rich in vitamin C. And that's why there are no native primates except for humans there. And we need that vitamin C. So that's an interesting story. There's another one, a little shorter, I promise, of a very important immune system protein called theta defensin, and we don't have it. So you may have never heard of it because when I say it's important for the animals that have it. And it protects against retrovirus infection. Well, humans don't have theta defensin. We have the gene, but it's been mutated and it's a pseudo gene. It does not express at all. And if we did have it, it's very likely to assume that HIV would be unsuccessful in infecting us. So the entire AIDS epidemic could be avoided, could have been avoided if we didn't lose theta defensin in this. So baboons have it, lots of other creatures have it, and if we did, I bet we wish we did. And so these pseudogenes, these are just two famous examples, but we have literally thousands of these in our genome. In fact, one estimate is we have about 15,000 dead genes in our genomes. And most of them are just copies of other things that we do have functional copies of, but some of them, like gulo and theta defensin, we didn't have a backup. So when they got destroyed, they took all their functionality with them. And usually when it gets mutated once, unless you fix it quickly, it'll get mutated again and again and again. So it's mutated beyond repair. But sometimes if a pseudogene is very young, meaning it only has one or two mutations, sometimes it can be resurrected through a mutation that reverts to the functional form. But you have to catch it early, otherwise it continues to mutate.
And going back to your own book, Nathan, you actually did point out, and I quote, there was a danger of a very good chance that a pandemic could strike at any point.
I did say that in 2018.
And obviously, with the way that we live in densely populated cities and populations and global travel, I think you said something along the lines of, it's not difficult to imagine a doomsday scenario.
Yeah.
This scenario that you imagined, coronavirus?
Well, right. So this has been quite a cataclysm, but it could be much worse in terms of this virus it happens to be very contagious. It's not terribly lethal, but it is very contagious. And we just have very little natural defense against it. So it's running roughshod through our population now. But imagine if something that had the lethality of, say, Ebola. Ebola is not very contagious, but it is very lethal. So if Ebola were to get as contagious as coronavirus, for example, you could lose much higher percentage of the population. I was obviously not the first one to propose that we're facing a pandemic. The truth is we've been dodging this bullet for years. We have been dodging pandemics, and we just finally stopped getting lucky this past year. But we live in incredibly high population density, and our species never had this kind of population density until farming was invented. And that's only 15,000 years ago at the earliest, which means most of our evolutionary history, we lived in small bands. And when I say small bands, I mean 150 individuals or less and living outdoors. And so, if a new coronavirus, for example, which usually just causes cold symptoms, it would move through the population quickly and that'd be it. And it would be years and years and years before you dealt with another one. But we have such densely populated and large populations that these viruses circulate and they stay in the population for enough time to mutate and then circulate again. And you have these annual cycles. Influenza has been with us for hundreds and hundreds of years because it's basically a new version of itself every year. That was never the case in our deep past. In our deep past, we had much less worry about this kind of virus. We always had viruses and bacteria are always been a part of us, but this kind of infectious respiratory infections that move through and make you sick, this is something that is mostly a consequence of modern living.
To finish up our conversation, I picked out a quote from Nathan's book that comes from Lewis Thomas. The capacity to blunder slightly is the real marvel of DNA. Without this attribute, we would still be anaerobic bacteria and there would be no music.
What he's saying there is the simplest organisms that first developed DNA, if the copying of their DNA from one cell to another was perfect, they'd be, 4 billion years later, about 3.9 billion years later, it would still be those same cells, those same kinds of cells with no changes. So it's actually the errors that we make in copying our DNA that create new genetic material, new versions of things. And it starts off as a small version, small change, small fluctuation that then takes a life of its own. So what Lewis Thomas meant when he said that the capacity to blunder when copying DNA is what leads to all the diversity is that everything you see in any living thing is due to a slightly different version of its genes. So any variation from one individual to the next or one species to the next is just tweaks of the DNA. But those tweaks were accidents when they first appeared, right? So mutation is the process of changing DNA by accident. And of course, most of those changes are either neutral or bad, but we don't see those. What we see are the results of those rare but cool beneficial mutations that create new versions of things. And everything from us having 2 legs to us just having more than one cell. Because remember, most organisms on the planet have just one cell. So it was a mutation that allowed 2 cells to stick together as a unit rather than going their separate way. All of that comes from mutations, comes from what began as an accident. So that's what he meant. The capacity to blunder slightly is what makes everything great on Earth, and including music. But what he means by that is an ability that comes from a cognitive function, which comes from cells in the brain, which is got that capacity through mutations.
Thanks to Nathan for joining me. If you haven't read his book, then it's worth looking out for. In the next episode, I'll be joined by Professor Steve Hubbard and Dr. David Norman to take a closer look at evolution. Make sure you subscribe to the podcast on your preferred platform and follow us on Twitter @BiologyHire. Don't forget, you can get 40% off all Lecky textbooks, revision guides and practice papers using the discount code LeckyPodcast40 online at www.leckyscotland.co.uk.